Hello, welcome to the next A-level lesson from the conscientious biologist Ben Gallagher. This one's on prokaryotic ultrastructure. It follows on directly from the last video, which was just cell ultrastructure, but that one looked mainly at eukaryotic cells. This is a relatively quick one because there's not as many things that you need to know about in prokaryotic cells. But at the end of this video, I've put a table that summarizes the differences between the two forms of cell, because that's what most commonly comes up in the exam. As always, please do subscribe to my YouTube channel if you haven't done so already, and do follow me on Facebook and Instagram. Thank you. So you should remember back from the last lesson, or it was the first lesson, or from your GCSE knowledge of classification, that there's basically two major domains of life, the prokaryotes and the eukaryotes. Now, prokaryotes are split into archaea and eubacteria, but we still tend to lump them together as prokaryotes when we're talking about cell structures. We looked at eukaryotes in the last video, specifically plant and animal cells. Now we're going to look at archaea and eubacteria, but lumped together as one overall prokaryotic domain. So they've got to have the two essential parts that make them a compartment separate to everything else in the world. So that's the cell membrane, which is the kind of skin around the outside, and the cytoplasm, which is the liquid within, because that's where all of the little particles and chemicals are going to mix together, collide, and have their metabolic reactions. Now remember what a cell has to be able to do if it's going to function though. It needs to be able to make protein. And the two key things it needs to be able to, it needs so it can make proteins, are the genetic instructions for how to build proteins and ribosomes so that they can do the building. So let's look at the genetic instructions first. Well, bacteria do have DNA and we do call it a chromosome, but look how different it is to eukaryotic cells. In eukaryotes, we had those, be they single or double, they were still linear chromosomes. Linear meaning they had a start and an end. It was a piece of DNA. In prokaryotic cells, you have circular chromosomes. Doesn't mean they're a circle, it just means it's a loop. So you could kind of pick it up if it was on a much bigger scale, kind of shake it out, and it would be like a ribbon that's connected to its start. So it's as a loop, okay? The structure of that DNA itself is the same though. So if you're thinking about your GCSE DNA understanding of nucleotides and the AGCT, you might remember, it's the same basic code because the DNA code is called universal. All organisms on Earth have the same basic DNA code. It is arranged slightly differently in prokaryotes because it's not wrapped around proteins, but we can get to that in another video. Now, a key thing about prokaryotes is that they do not have a nucleus. In fact, they don't really have any membrane-bound organelles like mitochondria or chloroplasts or Golgi apparatus or anything like that. So there's no nuclear envelope here. But you do have a kind of paler region. You can see I've just made that paler. And what that's referred to in prokaryotes is a nucleoid region. That's where you find the DNA. Now they still have to have ribosomes because it's the ribosomes that are going to read the DNA, but hopefully you remember from the last video that in prokaryotes, they're slightly smaller. They're referred to as 70S ribosomes, that S being the Svedberg units, which is just a unit of size, okay? So slightly smaller ribosomes to read that DNA. Now, that's pretty much all a cell needs if it's going to function because it can make protein and therefore be metabolically active. But there's some extra stuff that they can have in prokaryotic cells that you also need to know about. So for example, a lot of prokaryotic cells have a cell wall. Now at A level, you need to know what that's made of. And it's made of something called murine, or sometimes referred to as peptidoglycan. Now you've got to know what it's made from so you can state it as being different to the cellulose cell wall that you find in plant cells. Fungal cells also have a cell wall, and their cell wall is made out of something called chitin. We'll deal with that in another lesson. But it's a murine or peptidoglycan cell wall, and for the same real purpose of a plant cell, that's to prevent it from bursting if it's in a watery environment. Water would move in by osmosis, but it can't burst the cell. It also gives a bit of strength and a bit of protection. Bacteria can also have a flagella, which is a projection that comes out of them. And within the flagella, there would be microtubules, because those microtubules can move. There's kind of almost like a little engine at the start of them, made of proteins that can waggle the microtubules around within the flagella to allow the flagella to move like a tail to allow prokaryotic cells to swim. Again, not all prokaryotic cells have these things, Some prokaryotic cells might have multiple flagella, but if they've got flagella, they're for locomotion, for moving around. Okay. A lot of bacteria also have plasmids. Those are just extra loops of DNA, kind of like mini circular chromosomes. You might remember talking about those at GCSE when we talked about genetic engineering, because you can modify plasmids within bacteria to get them to make gene products like insulin. Something else that they might have, going right the way around the outside, would be this green thing that I put on here. This is a slime capsule. This is made of a carbohydrate called mucin, which is exactly the same carbohydrate that you produce in snot. So they literally cover themselves in snot as a bit of protection, but also as a way of keeping themselves hydrated. Last thing that they can have sticking out of them at any place, they could be all over, they might only have a few, but they're little structures called pili. These are just little productions, little things that stick out of them, projections that they can use for attachment. So bacteria that might cause gut infections would have lots of these, because you swallow them, they want to stay in your gut, so they've got these little like grippy fingers almost that can grab on so they can stay in the environment that they want. So these are prokaryotic cells. Now eukaryotes, remember, are everything else. So that's animals, plants, fungi and protista, and we covered animals and plants in the last video. So take a screenshot of this. But at the end of this video, just like I did in the last one, I'll give you a blank of this that you can fill in yourself to practice in your own time. But take a screenshot of this one for your notes. Just to give you a better idea of what I mean, because the diagram, of course, diagrams, they are a bit restricted because we make it all really, really obvious for you. But if we were looking at some through a microscope, so here's a big bacteria on the screen here, you can still see most of the key things. You can see the cell wall is very obvious around the outside, and the cell membrane you can see is that dark line within that. You can see that paler region, that's the nucleoid. So even though we can't see it because it would be too small, you know that the circular chromosome is going to be within that nucleoid region. You can see there the cytoplasm. Cytoplasm you can label almost anywhere as long as it's not pointing to a very obvious structure. And within all that, those tiny black dots, they could be ribosomes, but you certainly know that ribosomes will be there within the cytoplasm. If I pull up another one over here, I've only really pulled this one up just to show something obvious. Yes, it's still got the cell wall there. Yes, it's still got cell membrane, nucleoid regions. The reason why you might have lots there is, of course, this is only a thin look through the cell. So the circular chromosome might be bent and comes up in different places. Or, of course, those pale regions may be where there's plasmids. Circular chromosome would certainly be within there, as would the cytoplasm and ribosomes. But look on this one, look all over its surface. You can see pili all over its surface. So this one would almost definitely be a gut-infected bacteria. I think this is Escherichia coli, which can cause food poisoning. So those pili would help it to grip on. You don't really need to take a screenshot of this one. That was just to try and help you to see how you can identify these structures if you were looking at them through a microscope. So let's look at the differences now. And as I've said before, this comes up in the exam all the time. So make sure you know this. Rather than asking you to draw one or label one, they quite often just ask you to give the differences between prokaryotes and eukaryotes. Normally they give it in the form of a table. So that's how we'll do it now. Now the most obvious difference is size. Prokaryotic cells are really small, normally on average between kind of 0.1 and 10 micrometers. So really, really small. We'll deal with the units of micrometers, nanometers, and millimeters in the next video on microscopes. But try and remember that prokaryotes are very, very small. Eukaryotes are generally 10 to 100 times bigger. So they're much, much larger cells in eukaryotes. There's no membrane-bound organelles in prokaryotes, where there is various membrane-bound organelles in eukaryotes. Just on that one, please don't make a mistake in the exam of putting, if it asks for three differences, don't put prokaryotes, no membrane-bound organelles, no mitochondria, no nucleus. That's not three differences. That's the same thing three different ways, okay? The nucleus and mitochondria are membrane-bound organelles. So if you said no membrane-bound organelles, saying no nucleus and no mitochondria is pointless. Those three would be worth 1 mark. So make sure you give distinct differences, and I would always put no membrane-bound organelles as a point on its own, then don't talk about the other organelles, okay, or there not being other membrane-bound organelles. There's a circular chromosome in prokaryotic cells. It goes around in a loop. Linear, meaning pieces in eukaryotic cells. Prokaryotes may contain plasmids, eukaryotic cells never do. This is a really important one, this next one. We haven't talked about this yet, and we'll do it in another video. But basically, whilst I said the DNA of prokaryotes and eukaryotes is the same, still the same code, same structure, complementary base pairing, double helix, all that stuff, in eukaryotic cells, the strands of DNA wrap themselves around big kind of round proteins called histone proteins. That's for protection and stability. That doesn't happen in prokaryotic cells. So for this table, please just remember prokaryotic DNA can be referred to as naked because it isn't clothed or wrapped up in these histone proteins. There's smaller ribosomes in prokaryotes, there's 70 Svedberg units, as opposed to 80 S in eukaryotes. The cell wall is peptidoglycan or murine in prokaryotes, whereas eukaryotes either don't have one, like our cells in animal cells, or the material they're made from is different. So in plant cells, it's cellulose. In fungal cells, it's chitin. So it's more about the material that the cell wall is made of that's the difference here. It's not presence or absence of a cell wall. It's what that cell wall is made of. Last one, but still quite an important one. Prokaryotic cells might have that slime capsule around the outside of them. Eukaryotic cells don't have slime capsules, but of course some eukaryotes can release exactly the same type of slime. For example, the cells in your respiratory system, they release that mucus, but for us, it's to trap prokaryotes so we can get them back out of our body. So take a screenshot of this because it's really important differences, and this does come up in the exam often. Now, as promised, and exactly the same as I did in the last one on eukaryotic cell structures, here's a blank of the diagram for you to practice on. Screenshot this, paste it onto a Word document, print it out as many times as you like, and you can just practice labeling all these key parts so you really understand the structure of a prokaryotic cell. That's it for this one. So from here, I want you to go on to the microscopy video. Now, the microscopy one covers quite a lot of stuff, but largely GCSE level recap with a few bits of A-level stuff layered on top, especially when it comes to the details of understanding resolution and electron microscope structure. So head to that one next. As always, please do like this video if it's been useful. Subscribe to my YouTube channel if you haven't done so already. You should just be able to click on the little circle of my face appearing above me. And do follow me on Instagram and Facebook. There's lots of additional biology on there to round out your knowledge and really useful for a synoptic paper if that's part of your scheme of work. Thank you.